Good evening to one and all present here. It's my voice are audible and PPT is visible. Okay, then we will start this session. So today we will be seeing week 5 live interaction session. So in this session, first off, we will revise the week 5 lectures followed by the sample MCQs. So myself, Ajit Kumar V, a PhD research scholar from Department of Bioscience and Bioengineering from IIT Guwahati. From IIT Guwahati. So today we will be seeing the week 5 live interaction session on the course Environmental Biotechnology for the course ID NOC 25 BT 83. So this session will be comprises of two things. First, we will revise some important concepts that has been covered in week 4 and week 5, followed by the MCQs. So before moving on to the course, I will introduce myself personally. Again, myself, Ajit Kumar, a JDP Joint Degree Program Research Scholar, which is a collaboration between IIT Guwahati and IIT BHU Varanasi. I work in the Laboratory of Biochemical and Environmental Engineering Laboratory, where my lab focused on working on the real-time wastewater problems across various industries, where my research inclines to the application of enzymes in the wastewater treatment, particularly at textile wastewater treatment. So moving on to my qualification, I have done BTech Biotechnology from Sastra University, Tanjavur, and I have done by MTech Industrial Biotechnology from NIT Surathkal, Karnataka. And about my work experience, I worked in many pharma companies like Biocon Limited and Zimbabwe. So this is all about my personal introduction, and we will jump on to the main concepts that has been covered in week 4 and week 5. So if you have any doubts, you can unmute the mic and you can ask your doubts. And if you are not comfortable, you can put your messages in the chat box so that I will view and I will clarify the doubts in and day. So I think we can move on to the lectures. So in the last week, that is week 4, we have covered some important concepts where we have seen the general aspects of environmental biotechnology, and we have seen different groups of microorganisms. We have seen prokaryotes, fungi, algae, protozoa, etc. And also we have seen the classification of microorganisms based on carbon source, energy, and electrons. And we have seen the combination of all of these two, followed by we have seen the importance of oxygen and how it helps for the survival of microorganism and how it is helpful in environmental biotechnology. And we have seen the classification of these microorganisms based on their oxygen tolerant level, where we have seen like obligate anaerobes, obligate aerobes, uh, we have seen facultative anaerobes and microaerobes. So these are all the some of the classification of microorganism that we have seen on oxygen tolerance. So after that, we have seen the importance of media, where we have seen the media culture, agar media, what kind of different kinds of agars that are being commonly used in the laboratories. Followed by we have discussed about it, the specialized media also, where we have seen enriched media, selective media, likewise, and we have seen what is the important criteria to choose a media also, and we have seen all these aspects, and we have covered the basics of media and their culture. Now we will continue with that. Here we will focus on growth of individual cells. So how cells is growing. So here we will be seeing three important concepts. One is growth, next is proliferation, and third is differentiation. So what do you mean by growth? Growth is nothing but a cell growth. Here we refer to the cell growth, where it refers to an increase in the individual cell mass or mass of the cell population. What does it mean? So the cells they grow or increase in their cell biomass, not in the number. Then what do we call if they increase in the cell number? In that case, we call it as cell proliferation or cell division. So cell division is nothing but there will be an increase in the cell number. As we could see, as we could see here, the cell growth refers to there will be an increase in the biomass. There is no increase in the number of the cells. Whereas if we see the cell proliferation, here the one cell is divided into three identical cells. So proliferation often refers to increase in the cell number. Example, binary fission. So in prokaryotes, what happens? 1 becomes 2, 2 becomes 4, 4 becomes 16. So there is always a 2 to the power of n division of cells occurs. So if there is increase in the cell number, we call it as a proliferation. If it is increase in the biomass, we call it as growth. And the third important concept is cell differentiation. What happens here? Here, the transformation of microbial cells occurs into specialized cells. So, what does it mean? Like one kind of cell is converting into another kind of cell based on their function, survival, and structure and their proliferation rate for their survival. So, these are the three important concepts in the growth of individual cells where we have seen growth, proliferation, and differentiation. Where if it is increase in the biomass means we refer to as growth. If it is increase in the number means we refer to as cell division. And if there is a transformation of microbial cells into some specialized cells, we call it as cell differentiation. So this is the three important concepts. And in the next slide, we will see some of the examples of this transformed cells. So the examples of transformation of microbial cells into some specialized cells, here we could see four kind of cells. One is endospore. They adapt various strategies like endospore, exospore, anabiotic cyst, nitrogen fixing cyst, and bacterioids. So first, what is endospore? The term itself says that the bacteria form the spore inside their cell. And when it is happened, when will it store its spores inside the cells? Whenever there is an adverse condition or unfavorable condition for their growth. So here we will see an anabiotic cell, they are temporarily not active. So it is not permanently dormant. It is temporarily not active cell with lower content of water, and it is covered by a thick envelope. Why it is covering with a thick envelope? In order to survive the adverse condition like starvation, dry environment, or it can be high temperature. So so this helps them to survive under the harsh condition. So this is like a nature's survival capsule that they form during their adverse condition. And the next concept is exospores. Exospores, the cells, the spores that are formed outside the cells, it is similar to that of endospore by its property, but they are not formed in the mother cell. The main function of these cells are to increase their survival and dispersion of cells in the environment. So this is the exospore. And what do we call as an anabiotic cyst? It is nothing but it is an enlarged cell with main function of increased survival. So what happens here? The cell get enlarges and thickened, mainly in order to survive unfavorable conditions like dry environment, high temperature, etc. So the last one is nitrogen fixing cysts and bacterioles. So these are also again the enlarged cell, but their main function is to converting the atmospheric nitrogen into ammonia group for the organic substance. So these are all some of the transformation of microbial cells into the specialized cells. Like humans, we humans adapt to different climates, right? So during rainy season, we used to wear raincoat or carry the umbrella, and if it is very cold condition, we used to carry the sweaters. Like so, according to the condition, these microorganisms also have their own strategy by having their spore accumulates inside the cells or exospore or anabiotic cyst or nitrogen fixing cyst. So these are all the some of the examples of transformation of microbial cells into specialized cells. So till now we have seen the basics of how the cells divides and what are all the important terminology that we need to know, like cell growth, proliferation, and transformed cells. We have seen some of the examples. Now we will see cell cycle. So this is another important concepts that we need to remember. So what is cell cycle? It is a sequence of events that occur in an ordered manner. So this cell cycle includes two events. One is cell growth and cell division. So it acts as a unit of biological time that defines the life history of a cell. So this defines the life history of a particular cell. So it is a continuous process that includes all significant event, right from different phases like interphase followed by the different stages like prophase, anaphase, which include the cell division part. So it includes all the significant event of the cell, ranging from DNA duplication and cell organelles to subsequent partitioning of the cytoplasm. So this process of cell growth occurs where the cell absorbs the nutrients and prepare itself for their division. And this occurs as we already seen, it occurs in various phases which are specialized for particular stage of the cell. And overall process and these steps is different for eukaryotic and prokaryotic. So as there are structural complexity occurs in their own system. And there are three main cycles that are involved in the cell cycle. One is chromosome cycle, second is cytoplasmic cycle, and third is centrosome cycle. So these are the three important cycles that occurs in a cell cycle. And we come across that there is a change in the eukaryotic and prokaryotic organism as they have structural complexity in the cells. Okay, so this is regarding the cell cycle, and we will see what are all the phases of the cell cycles that occurs in the upcoming slide. So the next slide says the life cycle of the cell cycle. And as we have discussed, the three important cycle, which is chromosome cycle, cytoplasmic cycle, and centrosome cycle. In the first cycle, where mainly the DNA synthesis occurs, that alters with mitosis. Whereas in the second cycle, where the main focus will be given to cell growth, that alters with the cytokinesis. and centrosome cycle where centrosome is divided so that it can be inherited and duplicated according to the two poles of the mitotic spindle fibers. So in each stage, they are regulated by various factors. And what are all the factors? There can be an stimulatory factor or an inhibitory factor. So this decides whether the cell needs to undergo division or only increase in the biomass. So these factors decides the cells whether it has to undergo division or it has to undergo dormant state. So these are all the factors that are governing the cell cycle. And these cell cycles are divided into different phases. Each of us is defined by various processes. So this is the cell cycle where various phases, as we have discussed, interphase, prophase, metaphase, followed by the anaphase, telophase, and a single cell is dividing into two daughter cells. I hope these are all the like basic concepts that you have been gone through in the elementary level. So we will here only focuses on the phases of the cell cycle, which is more important in the environmental biotechnology concept. So now we will move on to the phases of the cell cycle. So if we see the phases of the cell cycle, there are five phases, which is O gap 1 phase, which is called as G1 phase, followed by S phase, followed by G2, and finally M, where the cell division occurs. So, the first phase, which is G0 phase, or it is called as gap 0 phase of the cell cycle. What happens here? This is a period where cell present in a quiescent state or resting phase. So what do you mean by resting phase? So here the cell neither divides or grows, but they are metabolically active cells only, but they do not do any function. They just stay in the resting phase. So we call this resting phase period as G0 phase. So it can be considered as an extended G1 phase or it will be a separate phase out of the cell cycle. So what happens usually? The cell enters into the G0 phase when they mature, reach their mature, like in the case of muscle cells and nerve cells. What happens in the muscles and nerve cells? Once it has get matured or it has fully divided, these muscle cells and nerve cells remains in the gap 0 unless until it is mutated or it has changed due to external condition. So if there is in a normal condition, these cells normally exist in the G0 phase in lifetime. But the cells continue to perform their function throughout their life. That's why I said it is in resting phase, but they are metabolically active. They will perform the activity, but they are in the resting phase. So they do not divide. So this is why the nerve cells, once it is damaged, the nerve cells, we cannot revive it back unless until some like genetic tools if we use, we can regenerate them. But unless until in a normal cells, these muscle cells and nerve cells cannot be brought back. So that's why these muscle cells are usually in the gap zero phase after maturity. In some cases, however, the cell might enter into the G0 phase from the checkpoint in the G1 phase. So, what happens if the cells do not have their appropriate growth factors or nutrients, or if the cell senses that we don't have enough energy to carry out means, so it will not enter into the G1 phase. These cells remains in the G0 phase as there are the nutrition limitation or the lack of growth factors. And in G0 phase, the cell machinery is dismantled and the cell continue to remain in the G0 phase until there is a reason for the cell to divide. So basically, it explains the gap zero. It is normally regarded as an quiescent state or resting state, but they are metabolically active. So this is the cell phase. Unless until the cell doesn't want to divide, they will be remain in the G naught phase. And also in the second case, if the cells has deficient or lack of growth factor or nutrient, they remain in the G naught phase. They won't enter into the G1 phase. So this is regarding the G naught phase of the cell cycle. So the next is G1 phase, gap 1 phase, which is it is a part of the interface. What happens here? The cells prepare for the cell division. Here only the precursor or what are they will synthesize the materials like carbohydrates, proteins, whatever material that needs to be prepared for cell division, it will occur in the G1 phase. So a cell enters into the G1 phase after the M phase of the previous cycle. As we could see that after M phase, it will enter into the gap 1 if the cell needs to be divided. So a cell enters into the G1 phase after the M phase of the previous cycle, and thus it is termed as an first gap of the first growth phase. So what happens in this phase? There will be no DNA synthesis, but RNA synthesis occurs. Why? In order to
Hello,
produce.
sir. Sir, the PPT is not visible.
Okay, now it's visible.
Ah, yes, sir.
So we were talking about the G1 phase where this G1 phase is also a part of the interface. And the main function here is the cells prepare itself for the cell division. So what are all the precursor that are needed by the cells? They will prepare here in this phase. So once a cell after the M phase, it enters into the G1 phase. That is why it is called as first gap phase of the first growth phase. So what happens in this phase? There will be no DNA synthesis, but RNA synthesis can occur in order to produce the appropriate protein that are required for their cell growth. Okay, so this is considered as a time of resumption. That is, they collect all the necessary proteins, carbohydrates, and other materials in order to prepare the cell for their cell division. And the duration of the G phase, G1 phase is highly variable among the cells. And in some cells, it can occupy 50% of the total cell cycle time because it is a preparatory stage, right? So it takes some cells takes more amount of time, some cells take less amount of time. Whereas in rapidly dividing cells, the phase is entirely omitted. And the important in the G1 phase is G1S checkpoint where it checks if the cell is ready enough to proceed into the next phase called S phase. So what happens here? So in order to pass from G1, that is gap 1 to S phase, they will have a checkpoint here. And this checkpoint checks whether the cell is ready for their DNA synthesis or not. Whether the cell has produced enough proteins or carbohydrates and other materials that can handle the DNA synthesis. So here the preparation stage will occur and it will be checked by the G1S checkpoint phase. And there are enzymes there in order to check whether this event is happening smoothly. So at this point, what happens? Events like what kind of events? Reduction of any DNA damage, nutrient concentrations are performed to ensure that the cell has enough machinery to undergo cell division. So as we have seen in the G naught phase, it is a resting phase or in the quiescent phase, but they are metabolically active. Whereas in the G1 phase, what we have seen, it prepares their cells for their division. So it prepare all the precursor that are needed for the cell division. And the third stage is synthesis phase, which is S phase. Here what happens, it is also a part of interface where an important events like DNA replication and histone proteins will be formed. And these are two important events that occurs in this phase. And the process are tightly regulated. Why is it so? Because the replication process and the synthesis of this histone proteins in precise manner. If there is any mutation or changes in the DNA level, it will reflect in the it reflects the whole system and collapses, and it causes mutation in the organism. So this system, it is carefully monitored, and these are also monitored by their appropriate checkpoints. So the production of histone proteins and other proteins are crucial or important in this phase, as the newly replicated DNA molecule, they require histones in order to form nucleosomes, because the newly replicated DNA will be um, coiled around their histoproteins in order to form nucleosomes. And in order to enter into this S phase, there is an G1S checkpoint. As we have seen earlier, there will be G1S checkpoint where entering from the G1 to S phase, the cells will check whether enough nutrients are there in order to replicate the DNA. Once it has passed, it will enter into the S phase. And this duration is moderately long, and it occupies 30% of the cell cycle time. And what happens during this phase? During this phase, as we have seen, the main event is the DNA replication. So the content of the DNA will replicate or doubles in the cells, but the number of chromosomes will be remain constant, as the division of chromosome doesn't take place here. We will see in the next phase. Therefore, at the end of the S phase, each chromosome will have double the number of DNA with the double set of genes. So from this, we understand that mainly in the synthesis phase, the DNA replication and formation of histone proteins is taking place. And, and the fourth phase, which is gap 2 phase, or we call it as G2 phase. It is also a part of interface where the main role of this phase is they collect the nutrients and they releases their protein in order to prepare their cell for cell division. Earlier we have seen the DNA replication. Once it has been corrected, it will send it to cell division. It will send the cells to the next phase, M phase. So this is also like a preparatory phase for the M cycle. So where here the G2 is also a part of interface when the cell is still in the resting phase while preparing for cell division. And G2 phase is also important. Why it's so? Since in S phase the DNA replication is already done, it will check, this G2 phase will check whether any damage occurs during the replication process to ensure the cells in the proper condition. And if the cells are in not in proper condition, obviously mutation will occur and it collapses the cell cycle. So this is an essential phase that checks for mutation or any DNA damage to prevent excessive cell proliferation. What happens if there is an excessive cell proliferation? It will convert into a cancerous cell. So in order to stop and regulate them, this phase is one of the important phase. So here the DNA repair is a crucial step in the G2 phase, as it repairs the broken DNA that are present in the DNA strand during the replication. So the entry of G2 to M is tightly regulated, as why? Since since the next phase is very important where cell division occurs, and it is tightly regulated by different proteins and complexes. What happens if there is any DNA damage or insufficient nutrients is there? So the cell will remain in the G2 phase and it will not pass through the cell division. So this is the important of the G2 phase. So in G2 phase, the main role is it checks for any DNA damage and produce or prepare necessary proteins and complexes that are required for cell division. So this is the fourth stage, which is G2 phase. Next is mitotic phase. We call it as M phase, and this is one of the most crucial phases where the cell divides. The cell will be dividing into identical daughter cell. That is, one cell will be dividing into two identical daughter cells. And the most important event of this phase is that karyokinesis, which is nuclear division. What happens here? The chromosomes separate into two distinct cells. The mitosis begins with condensation of chromosome, which then separates and move to the opposite pole. So what are all the steps that are involved in this mitotic phase? First is the prophase, second is the metaphase, anaphase, followed by the telophase. So these are all the different phases, and once a cell enters into the M phase, it will have 4n concentration of genetic material by ending with two cells having 2n concentration of DNA. So this is the most important phase, and in the whole phase we could observe that the main phase, S phase, where DNA synthesis occur, and in mitotic phase the cell division occurs. And these two phases are like preparatory even for the next important steps. And we also seen in every step there is a checkpoint in order to observe the event carefully. So in the first checkpoint, what we have seen, whether necessary proteins and nutrients are there for the cells to synthesize the DNA. In the second stage to gap two, whether correct DNA synthesis has occurred or any damage or any mutation has occurred, it will be checked in the S phase. Whereas from the G2 to M, whether the cell has prepared its own cell like proteins and their complexes in order to carry out to the mitotic phase. And in finally the in the mitotic phase, the cell divides. That is, one cells will be converting into two identical daughter cells. So these are all about the important concepts regarding the cell cycle, and we have seen the three phases in the cell cycle, which is three important events, which is chromosome event, cytoplasmic event, and centrosome event. And also, we have seen the phases of the cell cycle where we have discussed the first G0 phase, which is nothing but a resting phase or a quiescent phase, but they are metabolically active. And, and the first stage, which is G1 phase, where they prepare their cell for DNA replication. And in the S phase, the DNA synthesis occurs. And in G2 phase, it again prepares the cells and checks for any damage in the DNA that are synthesized in the previous cycle. And the M phase, where finally the cells divides, and the one cells will be converting into two identical daughter cells. So these are all the cell cycle phases. And now we will see there are many level of coordination between the biochemical, physiological cell activity during the cell cycle. So we have seen in every stages there is a checkpoint, and checkpoint contains various enzymes and their complexes. So what did it? How did coordinates? It coordinates at different levels. The first level is the individual RNA and enzyme synthesis and their degradation. And second level, there will be a regulation of enzyme activity by metabolites and cofactors. And third, there is a regulation of catabolism and energy storage. And fourth is the regulation of whole cell activity by different cell regulators. So these are all different level of coordination that occurs between the cell activity and the biochemical events during the cell cycle. Now we will see the important concept of exotrophy and endotrophy and how it happens or how it is seen in the cell cycle we will be seeing now. So first we will understand the term cell exotrophy and endotrophy. In exotrophy, an external source of carbon is converted into energy or the energy that is stored will be converted into carbon store. What do you mean by it? It will be stored like in the terms of glycogen, starch or lipids. So what normally happens here in exotrophy? The cells consumes the external nutrients like glycogen, starch and lipids and they store them as energy reserves. So when this exotropy event occurs, especially in the G1 and the G2 phase, where the cells prepare its cells for DNA replication as well as cell division. So this is the stage where carbon reserves are stored for or energy reserves are stored during the exotrophy. Whereas in endotrophy, what happens? They accumulate the store of energy and carbon is utilized for DNA replication and mitosis. What happens here? The stored energy in the form of glycogen, starch, or lipids or energy, this will be utilized for DNA replication and mitosis. And we know that where does the DNA replication occurs? It occurs in the S phase and mitosis it occurs in the M phase. So in the S phase and M phase we observe endotrophy event where the accumulated stored energy and carbon is utilized for DNA replication and mitosis cycle. So this is the difference between the exotrophy and endotrophy where exotrophy where the cells accumulate or store their energy as the energy reservoir. Second, endotrophy, where what happens, the accumulated energy or carbon is utilized for DNA replication and mitosis. So, exotrophy, we observe the event in the G1 and G2 phase, whereas in endotrophy, we observe this in the S and M phase. So, if the environment is not favorable or during unfavorable condition, what happens? The DNA replication retains. If there is any unfavorable condition for DNA replication, what happens is it doesn't allow the cells to pass on to the next phase. So what happens? They retain the cell in the phases of exotrophy itself. Exotrophy we have seen it is G1 and G2 phase. An extended period of exotrophy, what happens? It leads to the intracellular accumulation, over accumulation of this carbon and energy resource. And the alteration between the period of endotrophy and exotrophy, it is regulated by CAMP concentration and there is a change in the membrane potential. So this is the period of exotrophy and endotrophy in the cell cycle. And we know that And we know that in exotrophy, what happens? The cells tends to accumulate the energy reserves. And we observe these events in the G1 and G2 phase. And in endotrophy, once this accumulated energy or the carbon, such as glycogen, starch, and lipids, they will be utilized for the either DNA replication or for their cell division. And where we have seen these events, we have seen these events in the S phase and the M phase. So how does it is controlled? How does it control? It is controlled by the concentration of CAMP and also if there is a change in the membrane potential, we could observe the events. So this is the important concepts of exotrophy and endotrophy in cell cycle. So till now we have seen how how the cells are grown, and we have seen terminologies like cell growth, cell proliferation. What is the difference between them? What is the difference? What is the difference between them? What is the difference between them? And we have seen like, hello Tanu, oh, can you unmute your mic? So till now we have seen the growth, growth, cell division, and what difference between them and how the cell differentiate. And we have seen the some examples and what are all the concepts behind it. Followed by we have seen the important concept of cell cycle. We have seen three events were chromosomal, cytoplasmic, and centrosome events, and the main phases of cell cycle, which is G0 phase, G1, S, G2, followed by M phase. So this is all about the phases of cell cycle, and we have seen how this coordination occurs between the biochemical and physiological cell activity. And also we have seen the period of exotrophy and endotrophy and their role in their cell cycle. So now we will move on to the growth curve. And do you have any doubts till now? And if you have doubts, you can unmute your mic and you can ask your doubts. If not, we can move on to the next topics. So I hope no one has doubt. We will move on to the next concept, which is growth curve. Growth curve, and a growth curve means it tells us the growth of the cells. That is how a microbial population's growth. And how does it measure? It is measured usually by plotting the log of the viable cells against the time, and this we call it as a growth curve or the growth cycle. And we will be seeing the graph between these in the upcoming slides. And due to its exponential growth pattern, as we have seen before, the one cell will be converting into two, two will be converting into four, so on. So due to their exponential nature, the growth curve of the microbial population is hyperbolic. And in batch culture, there is an initial phase of lag phase where we observe no growth, followed by rapid growth. We call it as an exponential phase. Then there will be a stagnant phase. We call it as a stationary phase. Finally, due to the death of the viable cells, we call this stage as a death or decline phase. Between each of these phases, there will be a transition period between these two periods. And we will see this graph in the upcoming slides. So as I told, the graph will be plotted between the log of living bacterial cells versus time, where we could see different events. First is lag phase, next phase is the log phase, third phase is the stationary phase, and fourth is the death or decline phase. And what do we call the transition phase? So this transition phase from the lag to log, this is one of the transition phase where there is another transition that occurs between the log and the stationary phase, and the third transition phase will be seen between the stationary and the decline phase. So four important these are all the four important events, that is lag phase, log phase, stationary phase, and the death phase. So we will see each phase one by one in a detailed manner. And the first phase is lag phase. What happens here? The microorganism adapt to a new condition when it is put into a new media. That is, when we are placing a cell into a new media or a fresh media, it takes some time to adapt or prepare itself, as we have seen in the G1 phase of the cell cycle, right, where the cells prepare itself for their DNA synthesis. Likewise, in the lag phase, a cell or the microorganism adapt to this environment, which is new condition, when we put them into a new media. So what happens here? The cellular metabolism speed ups, they expand, but they are unable to replicate, which results in decrease in the mass of the cell. So here the replication or the division occurs in the next stage, where here there will be no increase in the number of living bacterial cells. They just adapt to the environment. And the period of the lag phase is related to the organism's preceding state of growth. It takes microorganism longer to accumulate if it has an nutritionally poor media. So what happens here, if the media is poor or they do not have an appropriate media for a particular organism, the cell's adaptation period will be very long. What happens if they have a very long period? So efficient degradation or efficient growth will not be observed in this case. So it's best to have an nutritionally rich media in order to have a shorter lag phase. So as the organism begins to transduce the protein, the lag phase lengthens. So in order to shorten the lag phase, what we need to do? We have to transfer from a nutritionally poor media into richer media. If we put them into the richer media without any delay, we could expect a shorter lag phase. So from this lag phase, we could understand that this phase where the microorganism adapt to this new environmental condition. Once it adapted this condition, the condition refers to as is the media composition and the environment, which includes pH, temperature, etc. So once it has a good place to stay, their lag phase will be very minimal. So now we will see the next phase, which is exponential phase. Exponential phase, which is in this phase, the microorganism multiple rapidly and they divide. That is in an exponential manner. So their metabolic activity increases. So normally the binary fission DNA replication begins in the earliest. That is, one cell will be dividing into two cells will be dividing into four cells, and etc. And the amount of time that it takes to double in size over this length of time, we call it as generation time. So what do we call as generation time? Generation time is nothing but the time it takes to double the number of population. That is, for example, if there is two cells, so two cell becoming a four cell, if it takes some 10 minutes, so this 10 minutes is called as a generation time. What happens here? For every 10 minutes, the cell number one cell will be converting into two cells, and the two cells will be converting into four cell for every 10 minutes. So the period of time it takes for an organism, it varies. It depends upon the organism. If it is a bacteria, it will have a lesser generation time, and if it is a fungi or yeast, it will take a longer time to double, so their generation time will also be higher. So in the exponential phase, the main event is they multiple rapidly. Until that, it multiples rapidly until they have a sufficient rich environment and media composition. And what happens if it doesn't have the appropriate nutrient? Then it will enter into the stationary phase. So now we will see the stationary phase. So what happens in the stationary phase? So as the bacterial population develops along the growth curve, it consumes all the nutrients. What happens in the exponential phase? The microorganism consumes the nutrients and they will double their cells. So once it consumed all the nutrients, the cells tends to accumulated toxic metabolites, or it waste product, it will excrete the waste product or inhibitory compounds such as antibiotic. So if this all happens, it will change the circumstance of the media. Sometimes it can increase the temperature of the media or changes its pH, which is unfavorable for the microorganism. When an unfavorable environmental condition occurs, what happens? For bacterial growth became difficult, or they don't grow properly, or their growth will be stopped. So the rate of reproduction of the bacteria reduces until when the number of cells undergoing division equals to number of cells dying. So when number of cells undergoing division equal to number of cells dying, so at this time, at this point, these bacteria stops to grow or divide, and it becomes stagnant, and we call it as the stationary phase. So what happens here? The growth rate. Growth rate we will be seeing in the upcoming slides. What is growth rate? Growth rate is nothing but how the cells divides with respect to the time. How the changes in the cell concentration occurs with respect to the time. So here what happens? There is no change in the concentration of the cell concentration. Why it is so? Because the nutrients got exhausted. There will be an higher accumulation of toxic metabolites and waste materials, and sometimes inhibitory compound accumulation also, like antibiotics, which are all unfavorable for their growth. So the growth rate remains constant because the cell count is not increased. So when a stationary cell is shifted to a new media, it can quickly switch to the exponential phase and restart their metabolic process. Okay. So once a stationary phase, if a cell in the stationary phase, so once a cell, if it is in the stationary phase, we can quickly switch to the exponential phase when we give an appropriate nutrient media to the compound. Okay. So, and the last phase is death phase. What happens here? The bacterial capacity, they loses their cell division. Why it is so? Because of loss of nutrients and consequent accumulation of metabolic toxic products in the media. So once it has reached that stage, what happens? They completely stop their reproduction and they start to die when they are exposed to this unfavorable environmental condition. So the death occurs quickly and uniformly. So the cell concentration declines to zero. So what happens? The total number of cells, dead cells will be higher when compared to the living cells. When while some microorganisms are resistant to this condition because they could form endospores which allow them to survive in the adverse condition. So what happens here? Once once the number of dead cells is higher compared to the living cells, the death phase starts. So we will once again go through first stage, which is lag phase, where what happens? The microorganism adapt to the new environment. That is, there will be no increase in the cell number. They only sit and adapt to this new environmental condition. Whereas in the log phase, what we observe, there will be rapid increase in the cell number in an exponential manner. Why it is so? Since it has already adapted to new condition, they start to replicate it faster. And in the third stage, when there is an exhaustion of the nutrient, and if there is some accumulation of toxic products or any antibiotics, the rate of cell division is roughly equal to the set of dead cells. So it will reach a plateau in the number of living cells. There will be no increase in the number of cells. Only already existing cells will be active. Once there is no nutrient or accumulation of toxic is higher, then the number of dead cells will be higher when compared to the live cells, which causes quick death of these cells, which causes them to enter into the death phase. So this is the four important stages in the growth cycle, growth curve of of a microorganism. So as we have seen in the growth cycle, we have seen one important concept called growth rate. Growth rate, we have seen, it is nothing but the change in the cell concentration with respect to time. So if you put into a formula, the growth rate is nothing but the amount of biomass produced to the time taken. And what do you mean by specific growth rate? It is nothing but the rate of production of biomass per unit initial biomass concentration. So if it is if it is considered the initial biomass along with the amount of biomass produced and time taken, then it is a specific growth rate. So the growth rate is nothing to do with the amount of biomass with which the process was started. It only deals with the amount of biomass that is produced at a particular time interval. So specific growth rate specifies the contribution of the initial biomass. It takes into the account of contribution of this initial biomass concentration. And specific growth rate, how it is calculated, as we have seen in the growth curve itself. So this is the log phase. If we take the slope of this line log phase, we could get the growth rate. The specific growth rate, we will discuss what happens to the specific growth rate at each phases. In lag phase, it is zero. Why it is zero? Because here no cell division occurs. What happens here? It is just adapting to the new environment, right? So if it adapts to the new environment, there is no division of cells. So the specific growth rate is zero in the lag phase. Then what happens to the stationary phase? In stationary phase also, there is no cell dividing. Already existing cells will be present. So since there is no division of cells or change in the cell concentration, here also the specific growth rate is zero. And what happens to the log phase? Log phase, what happens here? There will be maximum. The specific growth rate will be at maximum rate because the cell rapidly grow. As we have seen, 1 will be converting to 2, 2 will be converting 4 in an exponential manner. So the maximum we could see is in the log phase. And what about the death phase? And we know that in death phase, the number of dead cells will be higher than the living cells. So it will be in the negative or less than zero. So this is regarding to the specific growth rate at the different stages. So what happens here? The specific growth rate remains constant in the log phase, whereas growth rate continuously increases. Okay, so this is regarding the concept of growth rate and specific growth rate. If we see the growth rate, it is nothing but the amount of biomass that is produced per time taken. So at a particular time, what is the change in the cell concentration? Whereas uh, with specific growth rate, we could see that the amount of biomass produced is compared and consider the initial biomass as well. So this is the main key difference between the growth rate and specific growth rate, and it is very important for a microorganism growth. So if we see and if we see and if we see the mathematical relationship between the duration of G1 phase, cell cycle, exotrophy, and specific growth rate. And I think we have discussed earlier these concepts. What is G1 phase? Where G1 phase, what happens? They prepare their cell for cell division, for DNA replication, and cell cycle. We have seen exotrophy concept and specific growth rate. And what is a mathematical relationship between all these terms? We will be seeing now. So the first is the duration of the exotrophy. And we all know that exotrophy is nothing but where the cells consume the external nutrients and they store or accumulate as the energy reservoirs. And how do we calculate this? It is calculated by zero point 0.5 t minus 1. What is meant by t here? T is nothing but ln 2 divided by mu, which is nothing but the doubling time. And what do you mean by doubling time? Doubling time is nothing but the time taken to double the cells. For example, if I say in order to convert a single cell into two number of cells, if it takes some 5 minutes, then the doubling time is referred to as 5 minutes for the particular microorganism. So this is the doubling time and it is calculated by ln 2 divided by mu. And we know mu is nothing but the growth rate, and how it is calculated. It is nothing but the amount of biomass produced divided by the time taken. This can be this value can be obtained by plotting the graph between the log of number of cells divided by the time taken by the exponential phase. In the exponential phase, if you take the slope of this value, which gives the specific growth rate. First, you calculate the specific growth rate of a particular microorganism and substitute here, and you will get the doubling time. Once you have got the doubling time, you can put it in the this equation. This equation, which is for duration of exotrophy. Then you could get an idea of exotrophy. And in Tg1 phase, it is 0.7 T minus zero point 0.9. So if we rearrange this equation, we could see that this zero point 0.9 will be coming to the left hand side. So it is Tg1 plus zero point 0.9 divided by the zero point 0.7. So either way we can calculate, either if you know the duration of G1 phase, you could calculate the duration of the cell cycle. And if you could calculate the duration of the cell cycle, you could calculate the specific growth rate. So if you know any one information, rest all information can be obtained through these formulas. So this is the main mathematical relationship between all the parameter like G1 phase, cell cycle, duration of the cell cycle, and exotrophy, duration of the exotrophy or the specific growth rate. So this is one of the important concept, and you need to know these formulas for solving the MCQs. And if we see the next, the exponential microbial growth and their equation, first, as we have seen in the starting of the video, we have seen we have seen the concept of growth. Growth is nothing but there will be increase in the biomass concentration. And here the the biomass concentration is given by X. X is nothing but the biomass concentration. Whereas the proliferation refers to there will be increase in the cell number. That is why we have indicated by N. So what do you mean by balanced growth of microorganism? So there will be a proportionate increase in the biomass as well as the cell number. So if you see the equation, N is equal to N naught 2 to the power of N. How it is derived? If it is referred to a cell proliferation, we use N is equal to N naught 2 to the power of N. If you refer to the cell growth, we call it as X is equal to X naught 2 to the power of N. So first we will try to understand these terms where N is the number of viable cells, where N naught is the number of initial viable cells. N refers to the generation. Right. Likewise, X is equal to X naught. It is in the terms of cell concentration. How do we got this equation? So this is the change in the cell concentration by the time, which we call it as dx by dt is equal to mu x. If you try to integrate this equation, we will be obtaining this equation, which is mu is equal to ln x minus ln x naught divided by t. And how it is derived, we will see. I'll try to write with this pen. If it is not proper, you just tolerate with me. So first is we will integrate this. We will integrate this, which is dx by x equal to we will integrate. Here you have mu, right? So we will be taking mu outside because it is a constant. So we will put dt. Okay. So how this equation come? We are going to integrate this. We will be taking this x into this direction. So dx by x equal to mu dt. So what happens at the initial 0? 0 refers to at the initial stage. When time t is equal to 0, there will be some amount of concentration, right? We call it as x naught. And at the time t, that is whatever time you are going to run the experiment, and if we measure at that concentration, we call it as x. So this is the limit 0 to t, and this is the limit x naught to x. And if you integrate this, you will be getting 1 by x is nothing but the ln of, we have x and x naught, which is x divided by x naught, and equal to mu bracket. We have to integrate this. How to? It is t minus 0. So if we convert this equation, we will get this equation because ln x by x naught is nothing but ln x minus ln x naught. And here we have mu t. So we will be taking this t into this direction. So that's why it is under t. So here we will define the terms. X is nothing but the cell concentration at a given time. X naught is the initial cell concentration. And here t refers to the duration of the exponential growth, and this is how this derivation is derived. And if you see the specific growth rate of an heterotrophic bacteria, 0.2 to 1 higher our inverse, that is the bacteria has higher generation time or doubling time. That is why we could observe an higher a lesser specific growth rate, which is 0.2 to 0.1. Whereas in fungi, it is 0.01 to 0.2, which is very less, right? So what happens here, their doubling time is very higher. So it takes days to proliferate. Whereas bacteria, it takes about 24 hours to from some 48 or 72 hours. So this is how why the specific growth rate and growth rate concept is important. And you remember these formulas, it will be very helpful in solving the MCQs. So the next concept is growth yield and the growth efficiency. So what is the growth yield? Growth yield and efficiency, it is determined, the growth efficiency is determined by the term growth yield, and what do we call it as YXS, which is nothing but, it is a ratio of X is nothing but what? Biomass, right? So the ratio of produced biomass to the consumed nutrients. So this is y excess. If it is respect to the energy, we call it as YXE, which is the biomass produced by the energy consumed. So if we take the batch system, it is determined by y excess is nothing but xt minus x naught divided by s naught minus st, where the xt is nothing but the final biomass or cell concentration, biomass concentration. X naught is the initial biomass concentration, whereas S naught is the initial substrate minus ST, substrate concentration in the system. Why it is S naught minus ST? Because here we are providing the substrate, right? For example, I am providing 1 kg of glucose in the media and I will be adding the microorganism. What happens? The microorganism consumes this substrate. What happens if it consumes? Their value get decreases as the time proceeds, right? So, for example, if we take 1 kg of substrate initially and after 1 hour, what happens? Their substrate concentration might be dropped to zero point 0.5. That is why the initial concentration minus final concentration. As cells consumes these nutrients, their concentration decreases drastically as the time proceeds. So this is why and this is how the growth efficiency is calculated in terms of batch system, where it is given by xt minus x naught divided by s naught minus st. And this is one of the most important formula that you need to remember for solving the MCQs. So the next is for how about the continuous system without recycling of the biomass. So as we are given a constraint which is without recycling of the biomass, so the initial biomass will not be taken place, will not be considered here. That is why the formula has become yxs is equal to x minus divided by si minus se, because at initially we don't see that much cells, right? So that is why the final concentration, biomass concentration, and si refers to the influent, that is incoming substrate and outgoing substrate. Why it is incoming and outgoing substrate? Because in the continuous system, there will be continuous substrate addition and continuous product removal. So there is always the substrate will be present at the outside. Whereas if you see the batch system, once the substrate and cells are added, it will be removed at the end of the phase. So there will be one input, one output. Whereas here, continuous input will be there and continuous output will be there. So this is the main difference between the batch and fed batch, and how the growth efficiency is calculated in the both systems. So now we will see the both system, batch culture and the continuous culture. So what is batch culture? Which is I've already discussed that it is a semi-closed system where you will be adding the nutrient and you will be withdrawing the product. That is one input, one output. and supply of air, release of gaseous product, and addition of any additional substance like antifoams during the this cultivations are generally allowed. And what is the effect of growth phase? If there is an exhaustion of nutrients or accumulation of metabolites and biomass, it follows the same growth cycle, which is lag phase, log phase, stationary, and death phase. So it is typical growth cycle, and it is different from the continuous culture. And we will see how the growth cycle is observed in the continuous culture in the upcoming slide. So if we see the growth curve in respect to the batch system, there will be first initially the lag phase, and we know that this phase is where the cell adapt. They have control of their environment, and this duration is depends upon the inoculum that we are providing. If the inoculum is already adapted to that environmental condition, we could observe a shorter lag phase. And the second is shorter lag phase, or it will enter into the transition phase where the exponential phase or log phase will be carrying out. Then we could observe the transition period between log phase and the stationary phase where there is an exhaustion of nutrients is going to happen. And finally, stationary phase will be occurred, that is mainly due to the exhaustion of nutrients or accumulation of metabolites. Finally, the number of dead cells and their lysis will be outnumbered when compared to the living cells, and we could observe a death phase for this microorganism. So this is the typical growth curve that you will be observing in the batch system. And what about the continuous culture? In continuous culture, there will be a continuous input and we'll be taking the continuous output. So, and it is an open system, whereas in the batch culture, we state that it is semi or closed system. So here it is an open system with the environmental condition, which is maintained with continuous provided provision of nutrients and waste removal. So how the growth phase will be? The microbial will be maintained in the lag phase alone. Why it is so? In order to keep them viable, so that continuously the cells will be proliferated and it will be directed for product synthesis. So here the constant biomass concentration will be extended for the period of time. And this is the last slide, and we will be moving on to the MCQs. So this last slide is regarding what are all the different configuration and reactors that are commonly used for aerobic and anaerobic bioreactor. The first bioreactor that we will be talking about is chemostat. Chemostat is one of the continuous cultivation system where we will be keeping the dilution rate of the system constant. And what do you mean by dilution rate? It is nothing but the flow rate. The flow rate that you are maintaining through the bioreactor divided by the total volume of the reactor. So here we will try to maintain the F by V ratio constant so that the cells will be maintaining in the viable state, which is in the log phase. If the cells are in the log phase, they will be in the more viable condition. So that's why we will be controlling them by altering the dilution factor, which is nothing but the flow rate by the volume of the reactor. The next configuration is plug flow systems, where we will be operating. It is one of the continuous system where we'll be sending the continuous input and collect the continuous output. Then there are different configuration where we will be connecting series and parallelly these bioreactors. And the fourth is fixed bed bioreactor or retained biomass bioreactor. What happens here is we will send the flow of media into it where biomass either retained or it will be collected separately through membrane filtration or sedimentation. Then with recycling technique with plug flow or chemostat system, and the last is sequencing batch cultivation, which is a continuous cultivation. So these are all some of the configuration, and we will be discussing this configuration in the upcoming weeks of the environmental biotechnology course. So now you just have an idea what kind of configuration and what kind of systems are prevalent in the anaerobic and aerobic bioreactors or continuous cultivation. So this is all about this week recap. And in order to revise that, first we have seen the concept of growth, proliferation, and transformed cells. Then we have seen eh, important examples of these specialized cells, followed by we have seen growth cycle and phases of growth cycle, which is G naught phase, G1, S phase, G2 phase, and M phase. And we have seen their cell checkpoints as well and how it is regulated. Then we have seen the growth curve where we have seen the four important phases, which is lag phase, log phase, exponential phase, and death phase. And we have seen the important concept of growth rate and specific growth rate, and how this specific growth rate will be in all the four phases we have seen. Then we have seen what are all the different configurations that are available in anaerobic and aerobic bioreactor, and we have seen some mathematical modeling, mathematical formulas that are used in the growth curve concepts, where we have seen the growth efficiency, how to calculate the generation time, how to calculate specific growth rate, and many more. So this is all for the today's session. And can we move to the MCQ? Can we move to the MCQ section? So this should be an interactive session. So I will read the question. You can post your answers in the chat box, or you can unmute the mic and you can tell your answer. So this should be an interactive session so that you can post your answers in the chat box. And if you have any doubts regarding the concepts, you can put your message in the chat box, or you can put your message in the discussion forums that are available in the NPTEL portal of the Environmental Biotechnology course. So where our team will be addressing those queries. Do you have any doubts till now? Do you have any doubts till now? Can we move on to MCQs? So this should be an interactive session, so you can post your answers. So you can post your answers in the chat box. Okay, then I hope everything is clear. We can move to the MCQs. So first I will read the question. You can post your answers. So we will see the detailed solution after that. First question. In microbial wastewater treatment system, the reduced organic matter from the waste acts as option A, source of electrons and carbon. Option B, sink of electrons and source of carbon. Option C, sink of electron and carbon. Option D, source of electron and sink of carbon. Option E, sink of electrons and CO2. Read the question carefully and tell. In a microbial wastewater treatment system, the reduced organic matter from the waste acts as your answers. You can post your answers so that we can discuss on it. So in a microbial waste treatment system, the reduced organic matter from the waste acts as either sink or source of electrons and carbon, or sink of electrons and source of carbon, or sink of electron and carbon, or sink of electrons and CO2. Option A. Okay. Any other option? Any other answer? You can see the question. You just take time and answer the question. Option A. Option A. Ashwini and Nibedita as told. So okay, let's check the answer. Yes, it is option A. The reduced organic matter is nothing but it is a source of carbon and electrons, right? So what happens here? It acts as a fuel source for microbes, allowing them to oxidize it to generate the energy for their growth. So source is nothing but it is generally regarded as electron donors, and sink is nothing but it acts as an electron acceptor. So first you have to understand the concept of source and sink. Source is electron donor, sink is electron acceptor. So generally the reduced organic matter acts as an electron donor for both electrons and carbon. So okay, we will move on to the next question. Cultivation independent approach like metagenome analysis is a modern alternative to pure culture study. Which of these is an incorrect comparison of these two techniques? I will read the question. We have cultivation independent approach like metagenome, right? So it is alternative to the pure culture study. So from this option, which is incorrect comparison between the metagenome and pure culture? Option A, the pure culture has only one microbial strain. Option B, metagenome has multiple microbial strains. Option C, less than 1%. Greater than 1% of all the microorganisms can be isolated as pure culture. Option D, 99% of all the microorganisms can be obtained in metagenome. Option E, DNA of all the microorganisms can be obtained in metagenome. So from these five options, which is incorrect comparison between the metagenome and the pure culture study? You just see the question and answer. So, so what is the answer? Option C. Okay. Option C. Greater than 1% of all the microorganism can be isolated as pure culture. Okay, we will see the answer. But the answer is option D, which is 99% of the all our microorganism can be obtained in metagenome approach. And why it is so? First, in order to know that, we have to first understand the situation, understand the definition of pure culture versus metagenome. As we all know, pure culture, it isolates and studies the single microbial strain, whereas metagenome, it is a culture independent technique. So with the DNA of all the microorganism, we can analyze their microbial community. Microbial community. So we will see why other options are correct. So if we see the pure culture, as we have seen in a pure culture, we could at that time it is used for isolate and study the microbial strain, only one microbial strain. Whereas metagenomic, as we have seen, it will collect all DNA of a particular in a sample and allows the study for microbial community. And here, 1% can be isolated, like greater than 1% of microorganism can be successfully isolated by the traditional method as well. But 99% of all the microorganism can be obtained. It is incorrect comparison because in a microbial community there are millions of microorganisms. So if we could extract, unless and until we could extract the DNA of this microorganism, we could not achieve this metagenome. So that is why it is an incorrect comparison. Though, though it is possible to comparison for incorrect for comparison of these two techniques, this is the false comparison. And DNA of all the microorganism can be obtained in the genome by extracting sequencing, which includes it is an cultivation independent approach, right? So DNA can be extracted, but extracting 99% is very difficult, and it is incorrect comparison between these two techniques. It is true with respect to metagenomics, but in order to compare pure culture and metagenomics, this is regarded as a incorrect statement. Okay, but you have tried, but the answer is D. So it's good. You just try. We will move on to the next question. Which one of the following option is true for a comparison on aerobic and anaerobic microbial process for wastewater treatment? I will repeat, which is a true comparison, which is the true statement regarding the comparison of aerobic and anaerobic microbial process. Option A, anaerobic microorganism. Anaerobic microorganism grow faster than the aerobic microorganism. Option B, slow growing aerobic microorganism are preferred in waste treatment. Option C, aerobic metabolism yield less energy than anaerobic metabolism. Option D, the anaerobic fermentation growth is the fastest in treatment technology. Option E, nitrifiers and methanogens are aerobic. So which of these following options is true for a comparison on aerobic and anaerobic microbial process, especially for wastewater treatment? And your answer option B. Okay. Any other answer? You just take time. Any other answer? Which one of these following option is true for comparison on aerobic and anaerobic microbial process for wastewater treatment? Option B. Okay. Any other answer? Okay. We will see the answer. The answer is yes. The option B. Slow growing aerobic microorganisms are preferred in waste treatment. Why it is preferred? We will see. The slow growing microorganism, they are, tend to be more reliable and stable. And this stability is one of the important factor for considering in the wastewater treatment. So slow growing microorganism are preferred. And why not other statements? We will see. First statement, anaerobic microorganism grow faster than aerobic microorganism. No, it is false because aerobic microorganism will be growing faster. Anaerobic usually they grow slower because of their electron acceptor. And we all know the difference between aerobic and anaerobic process, right? Aerobic means anyone? Aerobic process refers to any process that occurs in the presence of oxygen, where oxygen acts as an electron donor. Whereas in anaerobic, instead of oxygen, nitrates and sulfates and other electrons will be acted as an electron acceptor. So generally, anaerobic microorganism will grow slower. And option C, aerobic metabolism yields less energy than anaerobic metabolism. This is also false because in aerobic metabolism, we all know that oxygen acts as an electron acceptor, right? So if you see, if you have gone through the lecture week 3 and week 4, we have studied about the redox potential of different electron acceptors, and among those, oxygen yields the higher energy than the other electron acceptor. So the aerobic metabolism often yield higher energy. Option D, anaerobic fermentation growth is fastest. No, anaerobic systems are normally they grow slowly, right? So it is not fastest, it is a slower growth. So slower growth, slower biomass accumulation will be happening. Option E, nitrifiers and methanogens are aerobic. It is partially true because some nitrifiers are obligate aerobes, but methanogens are strictly anaerobe. So this statement is also false. That is why the option B, slow growing aerobic microorganisms are preferred in wastewater treatment. So we will move on to the next question. The next question is new molecular tools for analysis of microbial community should meet four primary criteria. Which of these followings is not a one of these criteria? So I will repeat the question. There are four primary important criteria for developing any new molecular tools. But among these options, one is not one of the primary criteria. And I will read out the options. Option A, they should be inexpensive and simple to perform. Option B, they should reveal structure as well as the function. Option C, they should focus on eukaryotes and viruses as well. Option D, this method should be fast and high throughput. Option E, they should be able to separate the lead actors from the small contributors. I will repeat the question. Among these criteria, which is not a primary criteria for developing a new molecular tools. And your answers are answer. So I will show the questions. So new molecular tools for analyzing the microbial community should meet four primary criteria. Which one of these following is not one of these criteria? Among this, which is not the primary criteria? Okay, option C. Any other option? Any other answer? Any other answer? Okay, we will see the answer now. The correct option is option A. They should be inexpensive and simple to perform. If you have gone through the week 4 lecture, professor has discussed the main criteria for developing any new molecular tools. He has given four important primary criteria, and those are we will discuss. So first we will see the detailed solution. While cost and complexity, cost is this option is also an important criteria, but if you see the primary criteria, they are not included in the primary criteria because if you see these high-end molecular tools are they are more often expensive and they are more complicated than the traditional test. If, for example, the conventional qPCR is cheaper and simpler than the metagenomic analysis, but the amount of data that we are getting from the metagenomic analysis is very huge, right? So that is why it is not one of the primary criteria. It is the secondary criteria. But the primary criteria should reveal the structure as well as the function because we have to know what is their composition and their function and what they are doing in the microbial community. And second, they should focus on eukaryotes and virus as well because we should not target only the prokaryotic organism. It should examine the structure and function in all the organism. So this is how you have to develop new molecular tools, not only for prokaryotes, it also extends for eukaryotes and virus too. And we know that it should be fast enough and it should be high throughput. That is, It should handle large amount of complex data that should be very quick and very efficient. And they should be able to separate the lead actors from the small contributors, which means it should able to separate and distinguish our microbial communities lead actors from all other unwanted actors. This demonstrates the necessity of sensitivity and limit of detection and accuracy for any new molecular tools. So B, C, D, E are primary criteria, whereas A is secondary criteria. This is why the option A is not one of the primary criteria. So we will move on to the next question. When organic waste is disposed in a water body, the resident microorganism degrade it. Which one of these following is not a likely result? I will repeat the question. When organic waste is disposed in a water body, what happens? And the resident microorganism degrades it, right? So which of these following is not a likely result? Option A, provided the waste is biodegradable, the microorganism shall completely detoxify it. Option B, depending upon the waste composition, algal growth may be enhanced. Option C, introduction of allochthonous carbon is a natural process which is excessively increased by this waste. Option D, biodegradation depletes organic carbon content and releases CH4 and CO2. Option E, biodegradation enhances the oxygen content and drive growth of aquatic fauna. So, what is which among all this is not a likely result? You just read the question and try to answer. Option E. Okay. Any other option? You can put your answer. You can try out. You can post your answers in the chat box. Option E. Okay. Yes, the correct answer is option E. What happens? The biodegradation depletes the oxygen. Once it depletes the oxygen, what happens to the aquatic ecosystem? It damages, it kills the aquatic ecosystems like fishes, phytoplanktons, etc. So this is the wrong statement. And if you see the other statements, why it is correct, we could see, observe that since in the option C, they have said it is an biodegradable microorganism like heterotrophs or facultative anaerobes can break down these waste and detoxify or sometimes mineralize them completely. So this is correct option. And option B, depending upon the waste composition, algal growth may be enhanced. Yes, it is true because the organic waste contains what are all the compounds? Nitrogens and phosphorus too, right? If there is excessive amount of nitrogen and phosphorus, this causes eutrophication, which promotes the algal bloom. The algae temporarily increases the oxygen, but they die and decay. So, due to the presence of nitrogen and phosphorus, there is a chance of algal bloom may be enhanced. So, this allochthonous carbon, option C, the organic matter, what do you mean by the organic matter is introduced externally from the aquatic system. So, the waste discharge represent excessive carbon loading beyond the natural level. So, this is also true. And if you see the biodegradation depletes the organic carbon content, yes, what happens? The microorganism oxidizes the organic compound and it converts CH4 and CO2 in the case of anaerobic condition. So this is also true. And last option, as we have seen, it depletes the oxygen content. So once the depletion of oxygen content, it kills the aquatic fauna. So the option E is not likely a result. of the above statement. So we will see the next question. Total DNA-based metagenomic approach are not adequate to gain a complete understanding of microbial community because all genes cannot be detected, unculturable microorganism cannot be detected, all genes are not expressed. Option D, all metabolites cannot be tracked back to genes. Option E, all genes are not protein coding. I will repeat the question. Total DNA based metagenomic approach are not adequate to gain complete understanding of microbial community because it is total DNA based metagenomic approach. So which is why it is not able to gain a complete understanding of microbial community. Answer? Any answer? I will repeat the question. You can take time. Total DNA based metagenomic approach are not adequate to gain complete understanding of microbial community. You can post your answers in the chat box. Option C. Okay. We will see the answer now. Yes, it is correct. Correct, Ashwini. The option C is correct. All genes are not expressed because mere presence of genes as detected using the metagenomics doesn't guarantee their expression. We can identify the gene, but if it is expressed or not, we cannot identify through DNA based metagenomics. So how do we measure or guarantee this expression? We have to use other omics data or omics techniques like transcriptomics, metabolomics, proteomics in order to gather more data on this expressed gene. So here we could identify the genes. Genes can be identified. And as we all know, the metagenomics is nothing but it can identify without any culturable. It is a cultivation independent technique, right? So this is also true. But all the genes that are identified, whether it is expressed or not, we cannot identify or we cannot get these details from total DNA based metagenomic approach. So the correct option is option C, all genes are not expressed. Okay, we will move on to the next question. Microbial matrices are dirty, making extraction of cellular material difficult. Here, dirty refers to option A, presence of interfering chemicals. Option B, presence of soil particles. Option C, presence of other microorganism. Option D, presence of eukaryotes. Option E, presence of extracellular DNA from the dead cells. I will repeat the question. You can post your answers in the chat box. Microbial matrices are dirty, making extraction of cellular material difficult. Here, what do you mean by dirty? Whether it means the presence of interfering chemicals or presence of soil particles or presence of other microorganism or presence of eukaryotes or it is presence of extracellular DNA from the dead cells. Your answers are you can take time, look at the options. Microbial matrices are dirty, making extraction of cellular material difficult. What does dirty means here? Your answers are option B, okay. Option B, okay. Any other answer? Any other answer? Option A. Okay, okay, we will see the answer now. Yes, it is option A because here dirty refers to presence of extracellular enzymes and inorganic chemicals that damages the nucleic acid or prevent their effective extraction. Now we will see why other options are not. So the option B, the soil particle is also a part of microbial matrices, right? So here the dirty refers to the presence of various components within the matrix that are difficult to extract cellular material, which includes other organisms. If it represent organism, it can be an eukaryote or their extracellular DNA. So the correct option or correct answer is interfering chemicals. So we will move on to the next question. Lithotrophs involves the utilization of option A, organic matter as a carbon source, option B, inorganic matter as a carbon source, option C, lipid as a carbon source, option D, light as an energy source, option E, chemical as an energy source. So from this, what do we refer lithotrophs? Lithotrophs involves utilization of, from this five statement, you just choose what do we mean by lithotrophs. Any answer? Lithotrophs refers to, lithotrophs involves utilization of organic matter as a carbon source or inorganic matter as a carbon source or lipid as a carbon source or light as an energy source or chemical and energy source. Lithotrophs means option B, okay, inorganic matter as a carbon source. Okay, we will see the answer now. Yes, it is correct. Inorganic matter as a carbon source. And what do we call organic matter as a carbon source? Anyone? Like what do we call them? Organic matter. If it is used as organic matter as a carbon source, we call it as organic matter. Yes, it is organotrophs. If it is a lipid as a carbon source, we know that lipid is also belongs to the organic matter. So it also comes under the organotrophy. If it uses light as an energy source, it is a phototrophs. If it uses chemicals as general as an energy source, we call it as chemotrophs. So from this statement, lithotrophs directs to inorganic matter as a carbon source. So we will move on to the next question. Facultative anaerobes. You just observe the option and choose the correct one because all might have give a same opinion. So facultative anaerobes are option A cannot survive without oxygen. Option B they are killed by oxygen exposure. Option C they grow better with oxygen. Option D they grow better without oxygen. Option E they do not need oxygen but they are not harmed by it. So from these options, what do we refer to as an facultative anaerobes? Any answer? Any answer? What do we mean by facultative anaerobes? Option C. Okay. Any other answer? Facultative anaerobes. Okay. We will see the option. Correct answer is option C, as it is correct. Facultative anaerobes can grow faster by obtaining more energy from the aerobic metabolism in the presence of oxygen. Then, what about option A? Cannot survive without oxygen. If there is, it can survive. It cannot survive without oxygen. We call it as facultative anaerobes, right? And option B, they are killed by oxygen exposure. If there is a little presence of oxygen, they are killed. Then we call it as facultative obligate aerobes. Sorry, it is obligate aerobes. When it is grow better with oxygen, we call it as an facultative anaerobes. Though it might look similar to the option E and C, it needs oxygen for better energy yield. That is why we call it as a facultative anaerobes. So, okay. Then the next question is nitrogen fixing cyst formation. It is an example of microbial growth, option A. Option B, microbial proliferation. Option C, microbial differentiation. Option D, endospore formation. Option E, exospore formation. This has been discussed in the earlier lecture of today's also. So you try to remember which is the example for nitrogen fixing cyst formation, whether it is in microbial growth or microbial proliferation or microbial differentiation or endospore formation or endospore exospore formation. Any answer? Option C. It is a microbial differentiation. Okay. Any other answer? We will see the answer. Yes, it is option C. Microbial differentiation, and these nitrogen fixing cysts are formed via differentiation. So now we will see the other options, and we all discussed about these in the upcoming in the previous slides. Microbial growth is nothing but there will be increase in the cell biomass, whereas in proliferation there will be increase in the number of cells. Whereas differentiation refers to it will be differentiating from one state to the other state, right? Where it helps to this nitrogen fixing cyst helps to converting nitrogen, atmospheric nitrogen into ammonia. Exospore and endospore are nothing but formation of spores inside the cells versus the outside the cells. So the best example for describing the nitrogen fixing cyst formation is microbial differentiation. So these are all the MCQs that I want to discuss with you for this week. So thank you once again for patiently listening to this lecture. And if you have any queries regarding this lecture, you can mail me or you can put your queries in the discussion forum in the NPTEL course Environmental Biotechnology, where our team will clarify those doubts as well. And if you have any doubts, you can mail me personally at ajitvelu284@gmail.com. And if you have missed my earlier videos, you can watch those videos in the YouTube channel Ajit Kumar underscore V. Or if you could not able to find it, you can navigate to the NPTEL Environmental Biotechnology course, where you will have all these PPTs and the recording sheet as well. You can access every week's of the video if you have missed out anything. So do you have any doubts regarding the today's session? So do you have any doubts? So you can access the early videos in the NPTEL Environmental Biotechnology course tab. And if you still have any doubts in this, you can mail me or you can put your queries in the discussion forum. And today's session, we had discussed most important topic regarding the cell cycle growth curve, what all the formulas, what kind of different reactors that are used for aerobic and anaerobic bioreactor. And followed by, we have seen the sample MCQs for today's session. Okay, I hope there is no doubts. So we will wind up this session. And once again, I thank you all for patiently listening to these lectures and try to attend these lectures So we can discuss more sample MCQs which are very useful for your final exam for solving it. So thank you once again all. You can now leave the session and thank you. Thank you, Ashwini.